Hello students, in this video we will learn about the transcription method. So through this video I will discuss about the overview of transcription. That means in simple way you need to understand about the transcription method. So what do you mean by transcription? Transcription is defined as the process of information. It is mainly a process, and this process is mainly to form the RNA from the DNA molecule, mainly from its template strand. And it is the copying of genetic message coded in DNA into mRNA molecule. Actually, DNA contains information for the synthesis of cells specific proteins. DNA is located in the nucleate in case of prokaryotes and in the nucleus in case of eukaryotes, while protein synthesis occurs in the cytoplasmic part and mainly inside the ribosome, as you all know. DNA does not move to the site of protein synthesis to directly guide the process. Instead, it transfers its information to single-stranded mRNA, which move to the ribosome to direct protein synthesis. So transcription actually occurs inside the nucleus during G1 and G2 phases of cell cycle during interphase. Transcription starts at the promoter site and stops at the terminator site. Only one of the two strands of DNA duplex is transcribed. Since RNA is synthesized in 5' to 3' direction, it is transcribed only on DNA strand having 3' to 5' polarity. The 3' to 5', this one you can see, the 3' to 5' DNA strand that from which RNA is transcribed is called template strand, and its complementary strand having polarity 5' to 3' is called coding strand or sense strand, or it is also called anti-template strand. The coding DNA strand has the same sequence of nitrogenous bases as the RNA transcribed, except that in RNA T is substituted by U. That means thymine is substituted by uracil. So here each gene or set of gene contains a specific promoter region. Suppose this is the promoter site. This is the promoter site for guiding the beginning of the transcription method. And it is followed by the region of the genes that is transcribed and ends with a terminator. Here it is the terminator that mainly stops transcription method. DNA is unowned at the promoter site. You can see DNA part is unowned at the promoter site with the help of enzyme RNA polymerase. So this is the enzyme RNA polymerase that mainly unowns the DNA strand or DNA duplex, and only one strand of DNA, this one, this is called as template strand. So only one strand of DNA called the template strand is used to guide RNA synthesis by the RNA polymerase enzyme, and these strands runs in the 3' to 5' direction, in the 3' to 5' direction. In second stage, as the RNA polymerase moves along the strand, it adds complementary nucleotides. These are nucleotides. Suppose here it is A in that DNA strand, so here it adding U. If it is C, here in the for the mRNA molecule, in that place it placed G. So in this way it adds complementary nucleotides. So these are all nucleotides So it adds complementary nucleotides as directed by these DNA template strand, forming the single-stranded mRNA. Here you can see the formation of this single-stranded mRNA that reads in the 5' to 3' direction. So suppose this is the DNA template strand and it runs in the 3' to 5' direction, and the mRNA molecule which mainly form here, it mainly runs from 5' to 3' direction. So one of the DNA strand is mainly used as a template strand to form this mRNA molecule here. Last stage, that is the elongation stage, and lastly the termination step. The polymerase continues transcribing until it reaches a terminator site, and the mRNA transcript is released. So here the mRNA which mainly transcribed is mainly released for the formation of protein synthesis for the translation process. Note that the section of the DNA that has been transcribed is re-owned into its original configuration. So the DNA which mainly unowned previously by the DNA polymerase, that becomes re-owned into its original configuration. So this is about the overview of transcription. Hope you like the video. Thank you very much.